Okay, so welcome back. Now, after all these rather theoretical considerations about drones, we're going to talk about something really applied, and we're returning to the topic of mating control. Yeah, and actually, what I would like to present to you is a project that we had when I was still working at LIB, the Beekeeping Institute near Berlin, and which aimed at revalidating the Moonshine method of mating control. And the project went from 2019 to 2022, so it just ended basically. And it was a cooperation between two different breeding associations, one which works with Carnica and one which works with black bees in Germany, and our institute. Okay, so the problem we try to solve with this approach of moonshine mating is the same problem that you try to solve with your mating station, or that we try to solve with the insemination. So we have this comet of drones, we have this mass of drones in the, at the place where the mating occurs, and how can we control which drone will mate the queen? We have this is a problem that no other animal breeder has, but we have. If you have cows, you can probably control which bull will be on the meadow. If you have sheep, you can control. If you have bees, you have this particular problem. So we're worse off than everybody else, again. But we are the cleverest, so we will find a solution. So without mating control, the genetic progress is very slow, or there is no genetic progress at all. So this is a study. This is not actually a study, but this is a modeling. This is statistical modeling that has been done by Manuel Plate at our institute. And he actually modeled the genetic progress in years, depending on whether you have perfect mating control, so you can really determine precisely which drone mates which queen, or you have no mating control. And if you have no mating control, of course, it makes a difference whether the size of the surrounding population is very big or whether it is very small. So whether there is a lot of foreign drones coming in or whether there are few. But if there is a lot of foreign drones, then actually there is hardly any genetic progress at all. So we need mating control, and we know that there are limitations to the insemination, which is that it is very expensive and it is quite complicated to learn, as you all have first-hand experience. There's also limitations to the concept of the classical mating station, which is that there is not so many locations where you can do it. You're lucky because you're in the Basque country and there's a lot of mountains. But so that it would be good to have a third possibility. The principle of the moonshine mating is very easy. It is just to let out the drones and queens that you want to mate at later times. So you let them out only when you are more or less sure that all the surrounding drones will have returned to their hives. So natural mating normally takes place between 1 in the afternoon and around 5 in the afternoon, and moonshine mating will take place from around 6 in the afternoon and well, maybe approximately 7 in the afternoon, and then you close the hives again at 9. So this is not something that we invented. It existed for at least 100 years in different places, done by different people in very, very many different forms. And one that exists still today, and which was also one of our inspirations, was the famous train of virgin queens that Fanny Hagina put in place in Greece. Many of you will have heard of that. There's a nice YouTube video that everybody can watch. So in their case, what they do is that they store their mating nuclei in a cooled facility during the time of natural mating, and then they roll out the mating nukes on rails to the position that they where they will fly, where the queen will fly out. And then after the time of the moonshine mating, they will be rolled in again and will be cooled until the next afternoon. So this cooling is done in order to calm the colony so that the queen accepts to stay inside, and there is no loss of queens because they try to get out very hard and they die in the process. Fanny Hatina, the woman who built this in Greece, she came to our place and explained to us how she is doing, and we wondered together how we could simplify it a little. So our aims were to simplify the process, because you see, having a cooled facility is very expensive, it's very complicated to build. Bringing in and out the colonies all the time is also quite a lot of work. So we wanted to simplify, and also we wanted to check how pure the matings really are, which you can obtain with this method, because that has not been done very frequently. We tried to optimize the system for two different kinds of users. One would be small beekeepers. In Germany, we have around 95% of beekeepers who have very few colonies, so it's important also to do something for them, to allow them to control their mating. And we also try to have a bigger solution with a higher turnover of queens, which would be for associations running these structures. So I will start with the first part of the study, which was done for these smaller producers. And we actually cooperated with one of them, who was Mr. Skerka, who lives near Berlin. And he is breeding dark bees, which is really difficult in his place because all the apiaries around him have Carnica or Buckfast. So he's the only dark bee producer in the whole region. And of course, we wanted a system which is cheap, which is easy to replicate, and which is effective in terms of mating control, also if there's lots of foreign drones. So what did we do? We tried to automatize this process so that there is not so much manual labor involved with bringing out and bringing in these colonies. And the second thing is that we tried to find out whether this cooling that Fanny Hatina suggested was actually required. So we also tried without cooling. So in the experiment, we had one variant where we did the cooling because we did not know whether the cooling would be required. So we had one treatment where we did cool the nuclei, and this was the cooled variant. So the nuclei were standing here in this styrofoam cupboard, and these doors could be opened by just pulling them up like this. And behind these doors were all the nuclei. So nucleus, 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 nucleus standing behind these doors. And it was cooled with a household freezer. So this is an old thing that we had standing around in the institute, and we just connected it with a piece of tubing to the box and blew in some cool air into the box and controlled it with a little piece of electronics so that the temperature would be around 18 degrees in here. And then we had an uncooled version, and Egoitz has the uncooled version there. Yeah. So the uncooled version was actually based on the idea that the queen should not see the light while it is not allowed to fly. So while the queen stays enclosed during the time of natural mating flights, she should not see any light, because the light would just make her crazy. She would try to go towards the light and she would die in the process. So we would have to keep her in the dark. But it is important that the workers can fly, because if you put a nucleus in the sun and the workers cannot come out, you know what will happen. The nucleus will just die, right? So it is important to let the workers out somehow. So we had this special flight hole design, which consists in a double U-turn. So it's a kind of labyrinthum, we call it. So the workers, during the time of natural mating flights, the workers can go through this. This is an excluder grid. The workers can pass, the queen cannot pass, and the light cannot pass. And then at the time that the queen is supposed to fly out, we just put this to the left, and this hole will open, and the light will fall directly into the hive, and the queen will be attracted to the light and will fly out. This is what it looks in the action. Here you can see the queen coming back from a mating flight. And so the nuclei do not have to be brought into the cooled chamber and then brought out again. They stay outdoors all the time, and of course it's a lot less work. And then the change between these two flight holes, we try to do it automatically. So not have somebody do it manually, because it means being there every day at the right time, precisely the right time, and then again 2 hours later in order to close again. We did not want that. We want to open and close the thing automatically. This is how it looks in the real world. So this is now the worker flight hole which is open, and chuck, it changes. Now it's this flight hole which is open. And here's the queen coming out. Here it is flying.
Very quick.
Yeah. Mm. It's not always that quick, so.
Yeah. It's not so automatic.
Sometimes it takes 5 minutes or 10 minutes until she gets out. Yeah. And so this. Hmm? Should I play it again? Oops, sorry.
No, cierra. Aquí está abierto.
So we received a tip from this person here, Mr. Bast, who is a buckfast breeder actually in Western Germany. And he had worked with the Moonshine method before, and he said, well, to automatize this kind of thing, maybe try the motor from a car door. So you know that in the automatic closing system of your car, there's a small motor which moves the bolt. And so we took this motor, which is very cheap because it is an industrial part, so it can be bought for a couple of euros, and used it. And it does just that, well, this movement and this movement, nothing else. And we just put it there, and it seems to work. The thing that you see there, it can be done with a saw and some screws, and but it can also be produced by 3D printing. And in this case, actually, this labyrinthum is in this plastic block. So you do not need to put anything inside your nucleus. You just put the labyrinthum before the nucleus, and you only have to drill 2 holes, one here and one here, and attach the thing to your nucleus. And you can use any type of nucleus if you want. You can also use apidea. Of course, for the experiment to work, we also need drones, and the drones have to be regulated in the same way as the queens. So also the drones must not fly too early, because otherwise they will be home at 5 in the afternoon, and then they will not be available to mate our late flying queens. So we also have to regulate drone flight. So in the experiment, we had 4 colonies with the drones that we wanted to mate, which were dark bee drones, and we had around 30 buckfast colonies which were around 800 meters away in the apiary of a commercial beekeeper, and there was many other colonies around. So, but we only had 4 colonies of desired drones. That is important to know. Maybe it is a good point here to say that if you want to repeat this experiment, 4 is not enough. So it would be better to have 6, because in our case it worked well because the 4 colonies were really very strong and had drones all the time. But very often what you will have is one colony will lose its drones, or there is an uncertainty regarding the identity of the queen or of the drones or whatever. So it is better to start with 6 drone rearing colonies here. And it's really, then it works better. Because we had a couple of repetitions by now, so beekeepers who tried to replicate it. And so, well, one frequent problem was that actually the drone number was not sufficient after if they started with 4 colonies. So in order to regulate the flight of the drones, this is how we did. So these are 2 colonies standing side by side, and the actual colonies are here in this upper part. 1 colony and another colony. And these hive boxes that contain the frames, they're standing on an excluder grid, and underneath there is an empty hive box, just as you saw on the roof for preparing the drones for the insemination. So we have an empty hive box here just to create shadow, so to speak. And we have 2 flight holes, one above the excluder grid and one below the excluder grid. The one that is below the excluder, of course, will serve as entrance during the time when the moonshine mating is not taking place, and the other one will be for the moonshine. So we had our queens controlled, we had our males controlled, and of course we also had a controlled treatment. So we also had a number of nuclei where the queens were allowed to fly freely, so where there was no mating control. It was like a negative control, right? Mm-hmm. And so we had 24 of each that were used in the experiment, not all at the same time, but in 3 consequential rounds. And we had, in the control, we had 20 of the 24 that were mated, i.e. 83%. In the cooled, we only had 10 which were mated, which is not very good. And in the uncooled, we had 18 of the 24, that is 75% that were mated. So it seems like the cooling is reducing the frequency of mating success, so to speak. And we wondered why that is. And Mr. Skerka, the person where we did the experiment, he kind of gave us the reply, because he said that, well, these cooled queens, very often I see them leaving very late. So you open the hive, and it takes a long time until they come out. So we also had a couple of queens which were still there, but they were not mated. So apparently they were really not finding drones somehow. So an additional question that one may have is, if there are so few drones, just 4 colonies, and well, late in the afternoon, so that all the other drones are at home, do we actually have enough sperm in the spermatheca after the mating? So of course we dissected these queens and we counted the sperm in the spermatheca. And here is the number of sperm in the spermatheca. And here are these different treatments: control, the cooled and the uncooled variant. And what you can see is that there is some kind of trend towards fewer sperm in the spermatheca, especially in the cooled variant, but it is not very strong. So we always stay at, well, around 4 million, which would still be okay for 2 years of queen life, let's say 2 or 3 years. So actually it is still okay, and it is not very different from the control. Now what interested us most, of course, is the purity of the mating. Can we actually have mating control? And so because in this experiment we could not know what kind of foreign drones would be able to influence the result. We did not know what kind of foreign drones all the other beekeepers had in the surroundings, except for this one Buckfast beekeeper. We could not do any molecular analysis. So what we did was that we just compared the phenotype, the morphometrics of the offspring workers of these matings to the standard, the literature standard for Apis mellifera mellifera, right? And the two morphometrical characteristics which best discriminate between mellifera mellifera and Buckfast and Carnica, which is probably what was all around. We used the two most discriminating morphometric traits are the cubital index and the hair length. And so this is what we measured in the offspring of these queens. Okay, so here is the result of, or one of the results of this analysis. And these different colors here, yellow, orange, and red, they correspond to the 3 repetitions of the experiment. I told you that these 24 queens per treatment group, they were not all there at the same time, but in 3 rounds of the experiment. And the green bar here is for artificially inseminated queens. So these are sisters of these queens, which were artificially inseminated with the semen that we took directly from the drone-producing colonies that were there. So this is the result that would have been observed if there was perfect mating control. So this is the proportion of offspring bees from each of the queens that were not having the morphology that we wanted them to have. So they were showing a morphology which indicated that they probably did not have the fathers that we wanted. And you can see that with artificial insemination we're around 3%, which means that our drone-producing colonies were probably not totally pure. And with the control, that is with free-flying queens that were allowed to mate with any drones, we are around 25%. And with the cooled, we were around 5% here of this unwanted morphometrics. And with the uncooled, we are at around 12%, let's say, on average. So there is a lot of mating control. The mating control is much better than if you do nothing, but it is not perfect, of course. And yeah, what is maybe also interesting is that inside this bar or inside this group, there's quite a number of queens which actually appear to be totally purely mated. So there is maybe 30% of the queens which really only had nice offspring. So this is what we developed for the small-scale beekeepers. And now we also try to find a solution for a bigger setting with a higher throughput. And we did this experiment in an isolated mating station. Now, of course, you may ask, why are we doing this in an isolated mating station if the principle is to not need an isolated mating station anymore? And the reason is that in this place, we could actually control the number of the drones of the non-wanted genetics and of the wanted genetics, because only our drones were around. In this way, we also could do molecular verification of the results because we know all the genetics involved, and so we could trace them in the offspring. So also in this bigger solution, we had a cooled variant. And for the cooled variant, we did not build a small styrofoam box because we wanted it to be bigger to be able to treat more nuclei at the same time. And so we had a cooling trailer. So this is a commercial trailer that you can buy for refrigerated goods. If you transport cheese or milk or whatever, you can buy this kind of equipment. And we were lucky to have electricity in the middle of the forest because it is a place where the former president of Eastern Germany liked to hunt. So he brought electricity there so that he could have light while he shot his deer in the evening, and he had a pump in order to water the field which he grew in order to attract the deer and the swine. So we had electricity in the middle of the forest, which is really rare normally. So this was our cooled variant, and there was also an uncooled variant, and it was standing right here next to the cooled one. So this is the cooled variant, and the uncooled variant is here on the pellets. And these are the drone colonies with the drones that we wanted to mate. You know that ideally, as we discussed in the previous talk, these should be a little further away because the queens like to fly a lot. But we thought that probably in this moonshine situation where there is very little time and the mating flights are very short normally, probably it is best to have everything on the spot. So we really had them close to each other. Yeah. And the unwanted drones, they're standing here. So they're really standing not far away. And we had a little food for the bees, which was planted especially for us by a local farmer. That was nice. So these were the colonies for the drones that we wanted to mate our queens. And this is just to show you how it looks when all the drones come out and how it sounds also. So maybe you could hear it. It is really immediately the air is filled with this woo of the many drones. It's really like in a congregation area, very intense sound and completely different from the worker sound. And it is really like a bulb forming around these colonies with this sound everywhere in the air. And if you throw up a stone, then you will immediately see the drones flying behind the stone. And it's very nice. And these are the colonies of the drones that we did not want to mate, just to show you that they were there and they were also strong colonies. And now a very important question, of course, is to know when exactly to open and to close these flight holes for the queens and drones to come out. And so we supervised the presence of the naturally flying drones in our experiment. And we did that by, we first thought we could just observe the flight holes and say, okay, if there are no drones on the flight holes, that means that drone flight has stopped. But we noticed that actually drone flight is never stopping. So you can observe at 8 in the evening and still see drones. And okay, we said maybe not every drone that is flying is interested in mating, because drones also have to orientate, they have to train their muscles, they have to defecate. So we thought that maybe some of the drones are just there for these reasons. And so we tried to find a way to just monitor the drones which were interested in mating. And we did that by bringing up a pheromone bait, as you did in your mating station. And we brought it up with a little quadrocopter, which is very attractive to drones anyway, so even without the bait, interestingly. And we just counted the number of drones which accumulated after underneath the quadrocopter with a given concentration of pheromone and after a given duration. And we did that at different time points. Every 30 minutes we did that during the afternoon to see when the drones would stop accumulating. And this is one example. So this is an observation from one day in June of 2021, I think. And what you can see is this is at 2:30 in the afternoon. So this is really high time for natural mating, so to speak. And then it is slowly decreasing a little, the drone activity, but there are still a lot of drones here at 4:30 and even at 5. You can see quite a lot. And then there is a steep drop. And here at 5:30, there's almost no drones accumulating anymore. And this is quite repetitive. Everything that is here varies very strongly from one day to another. But this is relatively stable. We observed that if there is some rain in this period, then the drones tend to fly a little longer in the afternoon. So they try to catch up the time that they lost, and they fly a little longer here. We did not take this into account in the experiment, although probably it would be nice to do so for increasing the purity. And this is where we release the Moonshine drones, so the drones from our mechanism, and you can see that the drone number goes up, but then it goes down again very quickly. So this is at 17:47 and this is at 18:11. So there's not a lot of time for the matings to occur, for these Moonshine matings, right? This is the same time, but in minutes before sunset, because you were speaking of solar time. So as we found the optimum time to open this flight hole is, well, around here. So it would be like 265 or 270 minutes before sunset. Actually, we tried several variants, and we also tried different times. And the first thing we did actually was that we released them at the same time. And then we thought, well, given that the drones are coming out immediately and the queens usually need at least 2, 3, 5 minutes to come out, maybe it would be better to release the queens slightly before the drones.
Yes.
And so we tried that, but in the end, the results didn't vary very much. So for reasons of simplicity, I think it is totally okay to just release them at the same time. These are some results, and these are not all the queens that we tested. These are only queens which are comparable because all these settings were the same for all the queens in this graph. And this is the cubital index of 100 worker bees, 50 worker bees produced from each of these queens. And the green zone is actually what you would expect if it was pure mellifera. According to the literature, the mellifera should have a cubital index below 2. And as you can see, with the Moonshine method without cooling, most workers are inside this green zone, but there's almost always some workers which are not so typical of mellifera. And with cooling, it's about the same. So there's always 1 or 2 or sometimes 3 or 4 or 10 workers which are not really typical mellifera. So a lot of mating control achieved, but not perfect mating control. Actually, seems to have worked not so well as the first experiment that I showed you, but I think I can explain why in the next graph. In the two graphs from now. And then we also measured the number of sperm in the spermatheca again. And this is something I cannot quite explain. This is some apideas which we had. I didn't speak about it before, but we also had 5 or 6 apideas compared to these bigger mating nukes that I have here.
What is the name of this?
This is a Kirchhainer mating nuke. It's close to the Kieler.
This is Kieler.
This is not Kieler, it is Kirchhain, but it looks similar.
Is, smaller one.
Kieler is very similar to that one, actually. Yeah.
Apidea es más pequeño.
And apidea is much smaller, yeah. So we also had some apideas. We had the control, so those which were freely mated. And even in the control, we stayed mostly below 3 millions which was surprising given that there were so many drones around. And in the cooled variant and in the uncooled variant, well, doesn't vary so much. Uncooled, there's a lot of variation inside the sample, as you can see. There's also some unmated queens, apparently. But overall, it is between the control and the moonshine, there is not so much difference. The apidea seems to be a little lower. And it was very hot during our experiments. So our hypothesis is that actually they all suffered from the heat. And the mating was not so good because the semen suffered and did not enter the spermatheca all that well. This is our hypothesis. We cannot really say, but otherwise how to explain that even the controls, which were really mated under ideal conditions otherwise, had such a low sperm count. I mean, the apideas, they're much smaller. And well, we think that maybe the apideas suffered more because they were so small. This is the results from 21. In 2020, we also did experiments. And these experiments, we did not use the small mating hives, but we used full-frame mating hives. So like split boxes with 5 full frames. And there we had very good sperm numbers. So maybe this is also something that should be experimentally verified, whether actually these very small mating nukes can be a disadvantage under hot climates. I think we didn't do any experiments of that kind except this one, so I can't tell you. So we did a molecular verification of our mating control efficiency, and that was done by taking the semen from the spermatheca. So this time we do not work with offspring workers, but we directly work with the semen from the spermatheca, which is much easier because then you don't have the confounding effect of the queen. You only have the semen that you study. We created a small panel of molecular markers for mellifera and for carnica, which was suitable for our genetic material. And it was Dora Enriquez and Alice Pinto who helped us with that. And then we multiplied the DNA, of course, and then we sequenced these loci. I think it was around 40 different loci that we sequenced, and each locus was sequenced around 120 times in each spermathecal sample. So we get the results statistically from around 120 sperm cells, you can see. So we did one analysis per spermatheca, but from each spermatheca we did 120 repetitions of the sequencing, so to speak. So that is why probably this represents a number of around 100 sperm that were contained in the spermatheca. And now we have the results. And so this is a little difficult to explain. What is here is the proportion of alleles that are typical of the dark bee, right? So we did a mating in which we expected only dark bees to be produced, only dark matings to take place if everything went well. And as you can see with the free mating, there is almost no dark alleles in the spermatheca. So in the free mating, there was only carnica mating, which is not so surprising because it is a very isolated place. The few beekeepers that are there only have carnica because they're very strictly controlled by the local association. And so there are no mellifera drones around. There is only carnica mating. This is what we expect. It's a good control that our molecular analysis gives useful results. And actually, this is totally identical to what we obtain when we do the analysis with pure carnica semen. So semen that is not coming from a spermatheca, but it is taken directly from the drones, from these colonies. And if we look at our moonshine queens, this is the cooled variant. And you see that there's a high proportion of mellifera alleles in the semen, which is what we would expect. And actually, the median is at the same level with the pure mellifera semen. So if we take semen directly from the mellifera colonies and we analyze it, we found around 90% mellifera-specific alleles. So this is what would be perfect mating control if everything was directly on this line. In our uncooled version, it was the same, and this is another variant of the uncooled version where the timing of the opening was slightly different, but it is still the same. You see that there are some outliers here, so queens which did not work, which did not mate purely. The little stars also represent queens, so they are queens which are not mated purely, but overall the mating purity is actually very good, much better than the morphometrical results would suggest. And I think that this is due to the fact that here we use the real mellifera semen for calibration, whereas the morphometrical data that I showed before are compared to literature standards. So literature standards from 100 years ago, when Ruttner did his analysis of mellifera wings and hair length, and they do not correspond to the kind of mellifera stuff that we are using in the experiment. Okay, so some conclusions. Yeah, so we think that the Moonshine method works in general. There are some drawbacks, which is that the mating control is not always complete, and there can be cases where the sperm number in the spermatheca is lower than we would like, probably. Not always, but sometimes. But with regard to the effort, it actually compares very favorable to both the classical mating station and the insemination. So minus means that it is a disadvantage and plus means that it is an advantage in this graph. Because actually you only have to install the drone colonies that you want, and you have to install your nuclei, but you do not have to verify the genetics of all the beekeepers around you, as you would have to do if you had a classical mating station. And the equipment that you need is less expensive than for artificial insemination, and it is also easier to use, so it is not very complicated to apply. The cost, we think, is another advantage of the Moonshine thing. The mating purity is maybe the disadvantage. Of course, in terms of mating station, it depends on what kind of mating station you're comparing it to. So whether it is an island or whether it is mountain or just a flatland mating station. But we think that generally the mating control will not be as efficient as with a good mating station. Mating success is, well, we think probably not so much of an advantage of this technique. It reduces the mating success slightly, not so much if you use the uncooled version, but still a little. And of course, mating success is best with artificial insemination. And the really big advantage compared to the mating station is that you're independent of external factors. So you do not need an island. You do not need an isolated mountain valley. You can basically do it everywhere. And if you're interested in the system and just want to see a few more nice images, there is a movie on internet about this method that we produced last year with a very good documentary producer from Berlin, Mr. Thyssen, and yeah, it can be found under this link if you want. Yeah. There's also a movie about classical mating stations and another one about insemination if you want. So they're all by the same producer. We have a whole series. Last year we had a movie year, so to speak. Yeah. But I prefer the one about mating stations, actually. It's really nice. Yeah. So, of course, there was a lot of people working on this. Some of them are here. Thanks to them in their absence, and thanks to you. No, thanks to the funders, of course, and thanks to you for listening. Mm.